Um, Hello and welcome. I'm Shivan Sarna. I'm your host today for Kiran Krishnan. I created a community. It's called SIBO SOS. There's a great website, SIBOSOS.com, and I connect people with the information that's not usually talked about in the doctor's office. I hope that you will get inspired, that you have a lot of power that you can take and enhance your health through this information. Use your common sense. This is not medical advice. Instead, it is incredible information to help elevate your education and help you turn your learning into thinking. Kiran Krishnan is my special guest. He is the co-founder of Microbiome Labs, and he has probably done more studies in the United States, correct me if I'm wrong, on microbiome strains and research than anyone I can personally know. Kiran, you guys are so prolific with these studies and then the formulations. They don't have 1,000 formulations at Microbiome Labs. They have very specific strains that have very targeted purposes. And I really appreciate that. And he's one of the first people to talk to me about biological dentistry. Well, we nerded out that day years and years ago. And today we're going to be learning about how your gut can be helped by spore probiotics, by megaspore, as well as ZenBiome, as in Zen Cope and Zen Sleep. And one of the reasons, Kiran, I switched the topic to this one after our last session is when you told me about your daughter and how she was impacted by these formulations. So welcome. Thanks for being here.
Thank you. It's always a privilege to be here with you as well. And, and maybe people don't say it enough to you, but creating a community like you have is no simple task. I know you put your heart, soul, blood, sweat, and tears into it and have done for years. And thank you for doing that, because all the research in the world, all the work that nerds like me do will be meaningless if we can't get it to people within communities like the ones you created. So this is a critical partnership where we work together to empower people, right? So thank you.
My pleasure. Thank you. Right back at you. Everyone's invited to the Mutual Admiration Society, by the way. Membership is free. All right, we're going to talk to you about these topics today. And Kiran, I want to start with beginner's mind. There are a lot of people who've never been here before. What are spore probiotics like versus others? What makes megaspore so special? So it's just going to be beginner mind. What is up with megaspore and the role of sporebiotics in our gut?
Yeah, and keep in mind now, and I'll go into this as well, that not all spores are created equal, right? So we want to be able to distinguish that as we go along, because inevitably what happens in our industry is mostly made up of companies that are what we call copycat companies, right? So they borrow science, they hijack other people's science, they make products that mimic maybe to some degree what the science says. And then they go out there and they're largely focused on profits. And that's been something that, I've been rallying against for a long time. I just did an Instagram live with Dr. Nathan Bryan, from the nitric oxide company that he has. And that's the same struggles that he has, right? Because he's done so much of the research around understanding nitric oxide and how to formulate proper products. And his biggest frustration is all of the nitric oxide products that are on the market that do nothing, that don't deliver it, right? And these are all companies that are borrowing his science and the work that him and his team has done. So that's a really, really important thing. So I want people to keep that in mind as you're listening to it. Is everything I'm talking about is either work that we've been involved in or that we've done directly, and it's discoveries being made to elevate the industry, right? So we're trying to do the work that elevates this industry, brings more merit to it, and brings more, validity to the industry in general. So let's talk about spores and why we even went after spores to begin with. So probiotics are highly complicated, right? So what Shivan talked about, how there's a myriad of probiotics out there. They can be incredibly powerful and useful, but they're very complex because remember, they are living organisms. They have millions of genes. They produce hundreds of thousands of proteins. They create lots of different chemical reactions. They fight with each other. They work with each other. They fight with other things in the system. So it's not like a single molecule B vitamin, for example, right? That does a handful of things. You can predict what it does, and the metabolic byproducts of B vitamins are well understood. An organism is a very complex entity. And then when you mix multiple organisms, you're dramatically increasing the complexity of what that mix is and what it could potentially do. Many of you have probably seen the most recent Washington Post article and other articles that have come out that say probiotics don't do anything, right? This is the most recent article that came out. I had dozens of people send it to me and go, hey, what do you think? This article's title something like probiotics don't help or probiotics can hurt or something like that. And it's surprising to people that my response often is, I agree with the article, right? And the only thing I don't agree with the article is how they frame it, right? They say probiotics don't help. Well, probiotics is a big category,
Right.
That's like me saying, and they pull out a few studies on mixes of certain things that the researchers tested that didn't actually improve immune response, in fact, lowered immune response, mixes on things that actually did not recover the gut from antibiotics, in fact, slowed the recovery from antibiotics, right? Mixes of different strains. But those examples don't actually speak for probiotics in general, right? Because they're all so different. That's like me doing a study where I take, a couple hundred people and I make them eat fast food every single day, every meal for 60 days. And then I check all their blood parameters and I go, well, clearly they're much sicker than they were 60 days ago. And then I put out an article that says food kills humans, right? It's so general. What kind of food matters? So that's the part I don't agree with the articles, that it's for sensationalism. They generalize it like that. But there absolutely are probiotic products that can harm you. There are probiotic products that'll do nothing. And then there are probiotic products that will be incredibly beneficial if you use it under the right context, right? So that background is so important for people to understand. And this is one of the big missteps of even practitioners, is not understanding the differences between probiotics, right? And using ones in the wrong way and using, just these broad spectrum things with lots of strains and all that. That's a thing we've been trying to myth bust for like 10 years now. So let's get into spores. Why did we actually utilize spores and how did megaspore come about? Well, what we were trying to do, and what I always do, is try to mimic what happens in nature, right? The moment you do something that is wholly unnatural from how your body interacts with nature, natural compounds, and so on, there is a potential for it having side effects and harm, right? So when we look at a probiotic product, and let's say there's a probiotic product that has 15, 20 strains and 50, 100 billion CFUs of those strains, my first question I ask is, where in nature do you encounter that kind of mix, right? Is that a natural thing for our body to be coming across on a regular basis? And 9 out of 10 times, the answer is no. And if the answer is no, then there is a potential for something deleterious to happen, because that's not a natural process, right? So with that in mind, we said, okay, what is the most natural encounter of us with bacteria in the environment? And it turns out it's through the environment, right? Our ancestors ate dirt. They slept on the ground. Their food that they ate was covered in dirt. They drank waters out of rivers and streams. They foraged, they gathered, they ate animals' intestinal contents fresh after killing the animal, right? That's a known thing that hunter-gatherer tribes still do today. And in fact, that is the prize for the person in the hunting group that actually got the animal at the end, was they get to cut open the intestines and take the first bite of the intestinal contents, right? So that's the way in which we are naturally encounter microbes. It's through that environment. So then we started looking at the environment and trying to understand which microbes in the environment can actually act as a probiotic. What does that mean? Well, there is a accepted scientific definition for probiotic. Number one, it's a live microorganism. When administered to a host in adequate amounts, confers a health benefit to the host, right? Really important things there. Number one, for it to function as a probiotic, it has to be live and it has to be able to function alive, right? You have to be able to show that administration at a specific dose has a health benefit to the host. So there's a dose dependency. That's a very fundamental thing to understand the impact and efficacy of almost anything, right? Almost anything, vitamins, drugs, anything has a dose dependency study that is done so that you can see that A little bit of it maybe helps, a little bit more helps more, up to a point where there's diminishing return, and then you go all the way to the toxic level of that compound, right? So every compound, every nutrient and all that should have some sort of dose dependency ratio. That's what shows you that the compound is actually doing something in your system. It's absolutely true with probiotics as well. So it had to satisfy those criteria. So when we started looking at all the organisms that we could encounter in the natural world, it became very easy to filter down to a very small number when you look at definition part number 1, which is live microorganism. Because 99.99% of organisms we encounter in nature are going to get killed in the gastric system. They cannot survive through the gastric system because the gastric system is an antimicrobial system. It's called the gastric barrier for that reason, right? Many of them don't even survive the mouth because the mouth has alpha amylase, it has immunoglobulins, it has nitric oxide, all of these things that kill organisms the moment it encounters your mouth, right? In your salivary glands, you keep squirting out nitrate, which then certain bacteria in your mouth convert to nitrite, which is antimicrobial. That's a protection for us, right? So then we said, okay, there's a gauntlet there because your mouth kills bacteria, your stomach kills bacteria. If something gets past your stomach, your small intestine kills bacteria through the use of bile and pancreatic enzymes and so on. So there's a lot of things along the digestive tract that are designed to kill microbes. And so we said, is there a microbe that can actually naturally survive all that? If it can, that means it's probably intended to enter into your system alive, which satisfies step number 1 in the definition of probiotics. So as we started looking at the types of organisms, it became very clear very quickly that ones that could form spores are the only ones that can survive through this gauntlet, right? And when you look at the outside environment, the ones that can form spores, the most prolific kind are bacillus, bacillus being the genus, right? And then you have many different species. You have Bacillus subtilis, coagulans, clausi, indicus, and so on, right? So you have hundreds of different species of bacillus out there in the natural world, and they form spores. And that spore forming capability, and this is a bacterial spore, so don't get it confused by a mold spore or fungal spore, right? So a mold or fungal spore is like the baby of the mold. That's what is used to spread the mold all over the place. And then that becomes the progeny, if you will. And mold and fungus have a different cell structure than bacteria. They are different types of cells than bacterial cells. This is a fully functioning, mature bacterial cell that part of its life cycle, when it undergoes a certain kind of stress, will go into a spore state. That means that it becomes metabolically inactive. And it's a fascinating biological capacity. I'll give you what the human equivalent of this would be, right? So imagine this is a bacterial cell. It looks like this, right? You've got the bacterial DNA all over the inside of the cell. You've got the cytoplasm with all this liquid in it, and it's got all these little mechanics in there, like ribosomes and things to make protein. So if the cell decides that it wants to go into a spore form because the environment around it is not great for growing, what it does is it starts dehydrating itself. It starts sending out all the liquid, right? 90+% of every cell in your body, in the bacteria, in the yeast, in the fungus, in every animal is water, right? So it has to remove the water. And in fact, it's the water that causes cells to die under extreme heat or cold. Right? In cold, the water inside the cell will freeze, crystallize, and burst the cell. We know water expands, right, when it gets cold. Or if it gets too hot, the water in there can actually boil and increase temperature, and proteins denature, DNA denatures, and that's what kills the cell as well. So first of all, for survival, it has to get rid of the water, right? So it starts squirting out all its water. Then it moves all the genetic material to one side of the cell. And then what it does is it pinches off half the cell and sends it away. Then this side that contains all of the DNA and is heavily dehydrated will start forming a calcified protein armor-like coating around it, right? Now the cell has gone to sleep. It's in hibernation mode. It's no longer metabolically active. It can sit in this spore form indefinitely for up to 250 million years. Right? That's the oldest bacterial bacillus spore that's been found in a cave in Southern California in salt crystals. It was still alive. Putting it in the right condition, they could bring it back to life It was 250 million years old, sitting there, right? There was a fossilized spore that was found in South America inside amber, right? So what happened is a whole honeybee, an ancient honeybee, sitting there, got covered with amber. The amber fossilized, and you could see, like in Jurassic Park, they had the little mosquito in that piece of amber. You could see a whole honeybee in there, and so then they drilled in to get contents of the gut of the honeybee to see what it was pollinating, what it was eating at that time. They found a bunch of microbes, and the microbes, the spores, was still alive. They could still plate it and grow it. It was 50 million years old, fossilized in a honeybee, right? It's mind-boggling when you think about these organisms. So clearly, these organisms, and let me give you one more, this is the most mind-boggling of it all. The most likely candidate for establishing life on this planet, cellular life on this planet, are these bacillus endospores, right? There's a theory called panspermia, which is that the building blocks of cells, like nucleic acids and lipids and proteins and all that, were seeded on this earth, on planets that come from meteorites that bombard the earth, right? So because you find on meteorites, of course, you find all kinds of amazing elements, but you also find nucleic acids and cellular debris elements on meteorites that are floating around in the solar system, right? And so the idea was, where did the cellular life come, or the cellular building blocks for life come from on Earth? It was more than likely through the period of bombardment several billion years ago that brought proteins and brought nucleic acids and all that. And so then they started looking at, well, what could have survived? What existing today on Earth could have survived interstellar travel on a meteorite? And they did a study with bacillus endospores in space for 7 years, and it can survive at least a 7-year journey in space to come and bombard Earth with all the building blocks of life, right? So these spores are magical beings in many ways. They likely come from Mars, which is the most likely candidate. And they probably were the building blocks of cellular life, right? So you can't deny that looking into the spores is probably one of the most interesting and important things. So then we nailed down the spores. We narrowed down and said these things can survive the gauntlet through the system.
Can we just have a pause for a second? Because you just blew my mind. Okay, like I've never, you know how many conversations we've had? I've never heard you say that before.
It's mind-boggling.
It really is. Thank you for just like giving us that bigger picture, because I think we end up going like, oh, look, that's a pretty label. It's in a little, bottle, just like vitamin C at CVS right next to it, and just like Rolaids or whatever. But this is the power of what we're talking about. This is why so many people have their health transformed. Not everything's for everybody. I know that. So I'm not saying it's, a panacea, but I am saying it really has supported so many people. So I just wanted to mention that and just like have a beat where we go, what?
Yeah.
Oh my gosh.
It really cannot be overstated, I think, the importance of understanding the backdrop of all of this, right? Because it's not, and when you think about how we went through this process of identifying the correct strains and really understanding the importance of the strains, it's because of this kind of backdrop that, the kind of science that's been done that is available to us. And then how you think about the process, right? It's not just like, hey, my marketing guy said that, the last Google Analytics showed that spores are really hot right now. We should create a spore product. Oh, okay, great. Ring, my distributor. Hey, distributor, what spores do you have? Oh, I have 15 different spores that come from China and other places. Okay, great. I'll buy some kilos of those spores, mix it together and put it in a capsule, right? That is literally, that simplicity is 90% of what happens in product development in supplement companies, right? I know that because not only have I worked and supported other supplement companies, I've been in the room when these conversations are happening, right? About developing products and all that. And it irks me because this is what leads to the Washington Post article that probiotics don't work, right? Because people are putting things together without thinking about it. And I'll come back to a couple pieces of evidence around that. But so we went deep diving into what organisms in the environment are likely able to play that role as a true probiotic, right? Spores became the clear front runner. But again, like I said before, not all spores are created equal. Because spores are very ubiquitous, meaning they are found in every corner of this planet, right? They're found on the North Pole, on the South Pole, in the Tibetan plateaus, all over the Sahara Desert. There's these glacial ice core studies that have been done where they take cores out of really, really thick, miles thick glacial ice to look at what was in the environment, 50,000 years ago, 100,000 years ago, 2 million years ago. In every one of those environmental stages, they find Bacillus endospores as being very prevalent, right? So these spores are highly prevalent, but they're also really good at adapting to the environment that they're in, right? So a spore that you might find, sitting on a log somewhere in the woods doesn't necessarily mean that that spore can do anything in your body, right? Because it's now adjusted to living within the moss and the fungus and whatever organisms are there in that area, right? It hasn't adjusted to understanding the mucosa and what the host microbiome looks like and so on. So we said, okay, spores are critical, but we need to find human-derived spores. We need spores that come from the lining of the gut itself, because then these, we know, are the spores that are adapted for survival in the human gut. And so we work with the most prolific spore research institute in the world, which is Royal Holloway University of London. Dr. Simon Cutting, he's one of the most prolifically published authors on spores as therapeutics. And we work with him, and he had over decades identified and isolated human-derived spores that came from human intestines and human stool samples that he's properly characterized to species and genus, and he's tested to a certain degree, and we picked 5 of the top strains from him that were the most unique, that seemed to be the most prolific at their ability to colonize the gut and create metabolic changes. We licensed those strains, and then we started developing those. Now, the only reason we were able to get those strains from him, because him and the university controlled that intellectual property, was because we completely aligned on the importance of these organisms for human health. And we completely aligned on what we would do to show that, right? Our clinical program that we had in mind was absolutely aligned with what him as a scientist in this space would look at doing. This is why we won the licensing of these strains. We were going up against massive Fortune 500 companies for this, right? Their discoveries that they made through him, and something called the Color Spore Consortium, which involved 80 different researchers and over 8 million euros in research funding through the governments in Europe, were to discover these human-derived spores, especially ones that produce high levels of carotenoids in the body, antioxidants, right? That's our key strain, Bacillus indicus, that came out of that, which is found, of course, in megaspore. It's one of the 2 really, really key strains in megaspore. We were fighting against massive companies for that right? And we absolutely beat them purely because we were science first. I think what happened was that he became uncomfortable that some of these companies can just take it and just shelve it, right? Just so it doesn't get to the market. So that was a very fortunate thing. Then we started looking at what these do. Do they actually colonize, right? How do they impact the rest of the microbiome? What do they produce when they're in the gut? How do they impact the immune system? How does the immune system respond to the presence of these organisms? We went through painstaking detail of all that kind of work. And now Megaspore, which was the first all spore consortium product, which means it was the very first probiotic on this market that's all spores, 5 spores, right? Up to that point, there was only maybe 2 products on the market that had 1 spore in it, right? And then a bunch of other generic strains. This was the first all spore consortium product. 4 billion CFUs is all it takes. Doesn't require 50, 60, 100 billion CFUs. If you do the science and you know the science, you see that you're wasting a lot when you go 50, 60 plus. And then we started doing our clinical studies. We published our first study in 2017 on leaky gut. We were able to show in 30 days that we were able to alleviate leaky gut without doing anything else, without changing diet, without changing any behaviors and all that. A very, very significant reduction in translocation of endotoxins across the lining of the intestines, and then the inflammatory impact of that as well, right? So that was published in the World Journal of Gastrointestinal Pathophysiology. That was the first of, I think, now 13 studies that we've published on megaspore alone, right? So imagine in the last, so since 2017, it's been what, 6 years since then? In the last 6 years, we've published 13 studies on this one product, right? You've got companies, $100 million, $200 million companies that sell tens of millions of dollars worth of probiotics that haven't done one study, not a single one, right? In the decades that they've been selling these products. And so that's the point of differentiation, right? And like Shivan said, and we could go through the details of what all it does, but
yeah,
at the end of the day, here's how to think about it, right? This is foundational to health. This is why this became, by the end of 2019, became the #1 selling probiotic in the practitioner space, right? And we didn't have a marketing budget. We didn't have a marketing team. We didn't even hire our first marketing person until 2019, right? Because science is driving the bus for us. There was never a CEO for Microbiome Labs, right? Myself and my business partner, Tom, ran Microbiome Labs. Neither of us took the title of CEO because we didn't want an executive mindset or business mindset to run the business. I took the title of Chief Science Officer. He took the title of President and Clinician because he's a clinician and was for 25 years. We wanted science to drive the bus. We didn't want to make business decisions from a business perspective. We wanted to make any business decision from a science perspective only, right? This is why we never had a CEO. Every other company in this space has a CEO. And so that's a subtle thing, but it's important, right? Because that's what gets us to a place where, as small as we were investing so much into understanding what this product does, right? Understanding what these strains do in your gut. And I challenge you to ask any probiotic maker of product you've ever used, right? Call them up, email them, whatever, and ask them, hey, how does your probiotic impact the rest of the microbiome, right? See if they know, see if they have any data on that at all. How does your probiotic impact the immune system? And when I say probiotic, I mean the mix that they have, right? Not one single strain within their mix that may have some studies on it. What happens when you add them all together, right? What is that collective effect on the system? And we know that when you add strains together, it can actually knock out the effect. When we talk about ZenBiome, I'll mention the work that we've done there to see that. So it's so important. So think of megaspore as foundational. What we've done as a species is we've outsourced certain functionality to microbes because we don't have enough genetic material in our system to do all the services that we need to be human, right? We have about 22,000 functional genes. We have about the same number of genes as an earthworm. So we're not that sophisticated. The way we've survived and the way we're so sophisticated in our function is because we outsource a lot of functionality to microbes that live in our system. We have 2.5 million microbial genes in our system compared to 22,000 of our own human genes, right? So what we've come to discover is that likely a lot of microbiome maintenance is outsourced to organisms that we're supposed to encounter through the outside environment, right? And that's happened over the process of millions of years. It's called something called symbiogenesis, right? When you force two different species together, they will find a mutual benefit for working together. That's called symbiogenesis. They symbiotically, generate a relationship that is beneficial mutually to both species. And so us and microbes in the environment have developed this amazing symbiogenesis where we outsource some critical functions that we need in our gut, and then we give the organism a home where our immune system recognizes them as self and does not attack them, right? So what happens when the spores enter your system? They go in spore form. Remember, they're metabolically inactive at that point. When they hit the small intestine, they can actually bind to receptors on the lining of the mucosa, which awakens them and tells them that they're back home. That gets them within 8 minutes or so to come out of the spore form, rehydrate themselves, get their genetic machinery working, start producing proteins and start producing metabolic byproducts, right? They start working. Then they start interacting with the immune system. The immune system recognizes them as self. So what happens in general when they start interacting with the immune system is it increases the heightened awareness of the immune system to monitor the environment for other things. They increase something called toll-like receptors. Toll-like receptors are these detectors that are sitting on immune cells that the immune cells use to detect dysfunctional viruses, bacteria, foodborne toxicants, and so on, right? So the spores actually alert the immune system and go, hey guys, wake up, right? Turn on those detectors because you need to monitor the system, right? So it's a kick in the pants, to the kick in the butt, really, in the behind for the immune system to go, let's go, turn it on. And then at the same time, they can actually flag organisms in the gut that the immune system needs to pay attention to. Not only will they fire up the immune system, they'll go, hey, pay attention to this over here, right? This is a problem right here. You need to pay attention to it. Now, at the same time, they themselves can go after things that are problematic in the immune system, right? So, or in the mucosa in the gut. So bacillus produces lots of antiviral compounds. They produce antimicrobial compounds. They can go after viruses. So there's lots of studies on things like norovirus, influenza virus, that they can then shape how these viruses function in the body by competing against them, right? They can do that to other microbes as well, where they can compete against other microbes. We have 2 studies published through Cleveland Clinic on how these spores identify and compete against Clostridium difficile in the gut, right? And so that's just an example of how they can recognize dysfunctional organisms and start creating a competition for those organisms to try to balance back the microbiome. So there's lots of interesting functionality, right? They help digestion. They actually produce enzymes that are digestive enzymes in the gut to help you break down and assimilate food and nutrients, right? They, as I mentioned, they upregulate the immune system. They can also downregulate the inflammatory component of the immune system. They produce anti-inflammatory cytokines from the immune system, right? They also can improve the expression of tight junction proteins. These are the proteins in between each of the gut lining cells that tells the cells to seal up, right? Stop leaving big gaps in between, which is where things leak through. They rebuild the mucosal structure. And then to me, the best part is they increase the growth of other beneficial organisms within the system, right? They're really powerful at facilitating the growth of other keystone species as well. So all of these things I've talked about, those are all, tip of the iceberg in terms of what they do in the gut. But these are things, for the most part, we cannot do ourselves. We have, over the course of human evolution, outsourced these activities to microbes because we don't have the capability to do it, right? So what we've been able to work on and bottle into a product like this is decades worth of work by scientists to understand bacillus and understand bacillus endospores, so understand the role they may play, and then all the work that we've put into understanding them and the therapeutic role that we've played. So think about the hundreds upon hundreds of scientists, the tens of thousands of human hours that have gone into doing studies and understanding how things function and the benefits of it and how things interact and all of that stuff. All of that we get to bottle in a product,
amazing,
right? Because we participated in that process. And we were the first to bring this. And so that's really, really important. Now, again, I want to reiterate, not all spores are created equal, right? So before I started Microbiome Labs, I had a consulting kind of business where I'm working with companies on ingredients and product development and so on. And so when we first looked at the spores, right, my first strategy was I'm going to take it to these companies, right, and say, hey guys, these are incredibly important probiotic strains. In fact, I'd already formulated megaspore at that point with the right strains at the right doses and proportions, and we took it to several companies in our space, in the doctor space. At that time, this is back in 2012 and 2011 and 12, and they all laughed at it. Everybody rejected it wholeheartedly. They said, this makes no sense. Number one, nobody knows what the hell a spore is. Number two, spores are scary. People are going to think, oh, mold spore, you're going to kill us, Number three, they could not wrap their head around selling a 4 billion CFU probiotic. They said, hey, our best probiotic is 30 billion, 40 billion. How can we sell something that's 4 billion? And they could not wrap their head around the importance of it and truly understanding the science. And it's not about the billions and billions. It's about the quality of the strain, what the strain does, understanding all of that information. All of them rejected it, right? All of them. This is why Microbiome Labs exists, because if one of those companies did not reject it, this company wouldn't exist today. We would have just licensed them the spores, gave it to them, and let them do whatever they were going to do with it, right? But because all of them rejected it, because companies in our space just don't have that mindset or the innovation mindset, we decided to launch Microbiome Labs and bring this to market, right? Funny enough, every single one of those companies in the last 2.5 years have their own spore-based probiotic now, right? Every single one of them. How many of them have researched their spore-based probiotics? Zero. Not a single study has been done on any one of those products, right? It's just because we created a market, we brought the awareness of spores to the market. It became the #1 selling practitioner product, probiotic product in the marketplace. Now they see the market opportunity, and now they do what they do best, which is copycat, right? They, in fact, use our science. Some of these competitors actually use our research papers to promote their product, and the products are completely different. Right? So we have to be careful of that, because in order to support companies to continue to do this kind of work, people have to be very discerning in what they purchase. Because if you support those companies that do the knockoff stuff, don't do the science and all that, you're just lining their pockets and reinforcing that business model is the right business model, right?
Well,
If you only support, right, which is a choice you're making.
Well, then what also happens is you hear people say, oh, probiotics don't work. I didn't notice a difference.
That's my biggest fear.
That just makes me crazy. Listen, Kiran, we only have you for 20 more minutes, so I need to do some business here for everybody to housekeeping. Thank you all so much. One thing I didn't say at the beginning was be nice or be gone. So that is taken care of. Thank you. Clarissa, my dear, dear friend Clarissa, would you do me a favor and put the link so everyone here can participate in the special discount we have for Microbiome Labs? So it's right there. You click on that link. If you're not already signed up as a patient there, not patient, as a customer there, then please do so and use that coupon NoBloat15. And you do have to indicate that you are with, is it SIBO SOS is the client code? That's patient direct. Because of Kiran's relationship with us, we're able to get this to you directly. So we really appreciate that. It is a special dispensation by Microbiome Labs, and we don't take that lightly. So that's a great opportunity for you. There's also, for those of you that are new or weren't here last time, this is megasporebiotic for kids. And yay, it's a gummy. Oh my gosh, I have to stop myself from eating them, Kiran, because they're so delish.
Me too.
So
I had 3 today already.
I get it. So they have the regular, capsules, right? And then here it is in the gummies for those of us who are super sensitive, or you're like, I'm over the pills in general. I've already taken 10 capsules a day of something or other. So that's another option for you. What about, how do we take them? Do we take them with food?
Yep,
Before
always
we
take
go to
them with
bed?
food. Yep. And again, remember that we're trying to mimic what happens in the natural world. In the natural world, you would be consuming microbes with your food. Right? That's where the largest proportion of microbes will come in. So, and there's a relationship there, right? Because, for example, the spores require the presence of certain amino acids and some level of sugar to come out of that spore state and start functioning. So it's important to do it with food. The probiotic industry came up with some crazy things about, doing it in between meals and all that, because they were using microbes that can't survive the gastric system. And if you can't survive the gastric system, you're trying to avoid stomach acid, which is released when you start eating food, right? So that's why they said, oh, just take it in between meals. That doesn't make any sense. Where was it that our ancestors were able to carefully get their microbes into their system in between their meals, right? It just goes back to kind of basic foundational understanding.
Okay, so if you want to do the gummies, how many gummies is equivalent to 1 capsule?
So 1 gummy is equivalent to 1 capsule. So if you're an adult taking 2 capsules a day, just do 2 gummies. Most kids, my kids have been doing the equivalent of one capsule since they were probably 3 weeks old. So for them, one gummy is plenty. If things are going around, like at their school right now, there's lots of stuff going around, right? They will take 2 gummies a day, right? We would boost their exposure a little bit more, so which is good. So yeah, one gummy, great for maintenance, great for kids, great for sensitive people. Starting that way, 2 gummies is the full adult dose once a day.
Okay. Thank you for all that. We have to talk about the ZenBiome Cope and the ZenBiome Sleep,
Yep.
2 of the newest.
Yeah. So this is based on a strain called Bifidobacterium longum 1714. It's a wonderful story of how it was discovered. This was developed by our partners at Precision Biotics. As part of the Novozymes companies, we get to partner with lots of amazing companies that are founded by scientists, run by scientists. And this is one of those companies, right? It has a strategic partnership with University College Cork in Cork, Ireland. Which also houses the APC. The APC is probably the largest microbiome research institute in the world. So this product and strain was developed at the most prolific and largest microbiome research institute in the world, right? And there's 2 sister strains. There's one called longum 35624, which was developed for irritable bowel. That in itself has over 80 published studies on it. Right. And
Wow.
The 1714 strain is a gut-brain strain developed for mood, anxiousness, and then sleep as well. And this product itself the strain has 14 studies completed on it already, and there's more going on right now. So both ZenBiome Cope and Sleep, the only probiotic bacteria in there is the Bifidolongum 1714. Here's what's so unique about this organism. It has, like the spore has this unique spore-like coating, this one has a jelly-like coating called an exopolysaccharide, right? So it has this like crazy, elaborate carbohydrate tentacles all around it that covers it up. Right. And what's super interesting about the in-depth research that has been done on the strain is that it's been discovered that it is the exopolysaccharide. So this carbohydrate jelly-like coating that's what confers all of its benefits in a human host. Right. They've compared it to other Bifidobacterium longums, which you'll find in some probiotic products. It's a 1 gene difference. Right. Other Bifidobacterium longums are pro-inflammatory. They increase inflammation. And actually, we've done, and I've recently presented a lot of this work. They don't have any impact on the gut-brain axis at all. They don't impact cortisol at all. They don't impact brainwave activity. They do nothing for mood. They do nothing for stress. They do nothing for sleep. They actually amplify inflammatory cytokines in the body. And the only difference between those strains and this one that does all of those things, that does impact cortisol response, that does change brainwave activity, that dramatically reduces perception of stress, that dramatically improves sleep, the difference is 1 gene. And that gene in that entire bacterial genome codes for this exopolysaccharide, right? So this is why it's so important to know what microbes you're taking and that the microbe has research. Because if I said that Bifidobacterium longum has the effect of reducing stress and improving mood and improving sleep and changing brainwave function, I'll talk about the brainwave function part because to me that's the most mind-boggling part of it, and has this effect of bringing down inflammatory responses in the body, then people would go, oh, so Bifidolongum is important. Great. I'm going to Google Bifidolongum and you'll find Bifidolongums in many different products, right? But because those don't have the exopolysaccharide gene, they don't do any of those things. It's all the exopolysaccharide. So we've done studies where we've taken the 1714 strain, we've spliced out the gene that that codes for the exopolysaccharide, and we've grown the version that doesn't have exopolysaccharide and tested it. It doesn't have any of those benefits. In fact, we've been able to isolate just the exopolysaccharide And that by itself, even without the bacteria surrounding it, can have a lot of those benefits, right? So we know exactly the mechanism of action. So let me give you, and it does many different things, but let me tell you the absolute fascinating mechanism of action here, right? So what happens with this strain is when you consume it, it goes through, it survives through the intestines, it survives, sorry, through the stomach, through the proximal part of the small intestine, and then moves all the way down to the end of the small intestine. The end of the small intestine is this area called the Peyer's patches. That's an extra thin area of the intestinal wall, and there's lots of immune tissue there. Lots of immune sampling going on, right? In fact, dendritic cells and macrophages, which are these innate immune actors, are constantly going across and sampling what's happening in the system and what's coming through the pipe to train the immune system on what to be ready for, right? So that's the largest sampling site in the body. What happens when this strain tumbles around and is hanging out in the Peyer's patches, your dendritic cells actually reach across and consume the strain whole and pulls it into the lymphatic system, right? Now, what it does next is fascinating, because here's what dendritic cells normally do. Dendritic cells are called antigen-presenting cells. Their job is to eat stuff that's harmful, like bacteria, viruses, toxins, and so on, and then present some component of that bacteria, virus, or toxin to the immune system to tell the immune system, hey, this is what the enemy looks like. Form antibodies, form T cells against this, and go out and fight it, right? They are the first line of defense. They identify the problem, they present the problem to the rest of the immune system, and the rest of the immune system now knows what to go after. So that's what normally happens when they consume a bacteria or they consume a virus or something else that's disturbing the system. But when they consume this particular strain, it doesn't present any antigens to the rest of the immune system. So it doesn't activate the immune system in that same way, which is an inflammatory activation. What it does instead is within the dendritic cell, it digests the bacteria. It chops up the carbohydrates that are in the exopolysaccharide and the carbohydrates called peptidoglycans that are found in the cell membrane structure of this bacteria. It digests all those and it spits it all out into circulation. So the dendritic cells are actually delivering this unique component of this microbe to the rest of the body. And it is these components that have the anti-inflammatory effect. It is these components that prevent cortisol from making your gut leaky. It is these components that dramatically reduce the perception of stress. It's these components that actually induce sleep, right? And it is these components likely that change brainwave activity. So one of the most fascinating study, I mentioned I would talk about this at some point during this conversation, because to me, this is the most mind-boggling effect of this strain and the most potent, prolific effect of this strain, right? So there's a well-published study done by Professor Paul Ank in Germany, who's a world's expert on this whole thing of brainwave activity under stress response and all that, right? So your brain can have high frequency waves and low frequency waves. High frequency waves like gamma waves and beta waves are important when you're trying to do high focus activity, right? When you're in sports, for example, and you're running and you're trying to, your mind is really alert, or when you're in a project and you're trying to stay alert and you're reading through all these spreadsheets and all these things, right? That is a really active part of the brain. It's a high frequency wave that your brain goes through. If you encounter a stressor and you deal with the stressor in a high frequency brainwave, it amplifies the stressor and makes it much worse, right? That's the part of your brain that takes what could be a small, minuscule stressor and amplifies what the impact is. That's a part of your brain that spirals out of control, right? We all know that feeling. What if this happens, and what if that happens, and what if that happens, and so on, right? And you can't stop thinking about it. It just keeps going and going. Even at night when you're trying to sleep, it's going and going and going in your head. That's the beta and the gamma waves in your brain, right? What you need when you encounter a stressor is activation of the theta wave band or the alpha wave bands, which are the low frequency bands, right? These are the bands that are activated when you're in a meditative state. This is the benefit of meditation, is being able to tap into the theta wave, right? So the moment you encounter a stressor, your brain goes, turn off the beta waves and gamma waves, turn on the theta waves, because the theta waves allow you to turn on the coping centers of the brain. This way, you can understand the stressor for what it is. You don't spiral out of control. It reduces the cortisol and the stress impact of that stressor. Your perception of the stressor is completely improved. Like it blunts the impact of the stressor, and your body can go about without activating the flight or fight response, right? This is that calmness, that Zen that people are trying to reach through years of mindfulness practices, meditation, and all of that. This bacteria does that for you when you take the very first dose, right? This was the most fascinating thing that I saw in all of our research that we've done, is that this microbe can shift your brainwave activity during a stressor, right? That is fascinating when you think about it. The implications of that are vast, because most of us are running in this beta and gamma waves all day long, right? That's what makes you nervous about everything. That's what makes you nervous to get up and speak in front of a crowd. That's what makes you nervous about the deadline of a project. That's what freaks us out when we feel like we're running late, right? And that's what panics us about this one thing this person said and that other thing. And what if this happens and what if that happens, right? That whole mindset is beta and gamma waves. Being able to tap into theta waves and alpha waves negates all of that stuff, right? And it brings down inflammation in the body, because the beta waves and gamma waves that create all those thoughts are super high inflammatory waves, right? They create massive amounts of inflammation in your brain, in your heart, in your muscles, because they turn on the flight or fight response all the time, right? So if you want to be Zen, you want to be in a good place, you want to be balanced, you want to not have all that inflammatory damage that occurs from just dealing with life and life stressors and so on, you need to be able to tap into the theta and alpha waves, and the probiotic does that for you. So absolutely fascinating stuff. Maybe we have a little time for questions. Oh, I can't hear you, Shivan.
Thanks. I only have 5 more minutes with you. Kiran, I want to thank you very, very much. We're going to try to cram in a bunch of questions here.
Yeah.
I have so many. The strain that's being talked about with the theta waves and that kind of coding is the Zen Cope, right?
And the Zen Sleep, both of them.
And the Zen Sleep.
Yeah.
How exactly do we take these, Kiran? And
Yeah.
with the Zen Sleep, I'm not eating right before I go to bed. So how would I handle that?
So you don't have to take those with food. In fact, you could take them without food, totally fine. Because what we really want is get those strains to the dendritic cells in the Peyer's patches, right?
Okay.
So it's not important for those strains to work with food and do all that like the spores do. This is a different mechanism, and it's very specific to gut-brain function, right? So if you have bloating, this is not the strain for you, right? This is not what you use it for. You use this for stress, mood issues, and sleep issues. You use it with kids, you use it with adults, and so on. So the way the two products work, they're both 17-14, right? Most people who have daytime stress issues also have nighttime sleep issues, right? So if you have stress and sleep issues, what we really want you to get is about 10 billion CFUs of the strain. So the Cope has 5 billion CFUs per capsule, but it's also formulated with B6 and B12, which actually helps some of the beneficial mechanisms in the neurological system. And it's also formulated with saffron and lemon balm, right? And those two are very calming on the nerves. Saffron, in fact, improves GABA and a number of neurotransmitters that are really good in calming to the neurological system, right? So it's a great pair with what 17-14 does. So if you have daytime mood and stress issues, you take one Cope during the day. And if you have nighttime sleep issues, then you take one sleep at night before bed, right? Maybe an hour or so before bed. Now, the sleep is the same 17-14 strain, but formulated with L-theanine. And L-theanine has a lot of positive impacts on sleep efficiency and so on. So you combine the two. We combine the two to have supportive mechanisms.
And this has the lemon balm in it too.
Oh, sorry, that's the lemon balm. Yes. The saffron is in the Cope. So if you have daytime issues and nighttime issues, you take one Cope during the day and one sleep at night. Let's say you only have daytime issues. You seem to be okay at night. You found a ritual that helps you sleep. Then you would actually take 2 Cope during the day, right?
Okay.
Because that's what will give you your 10 billion CFUs. If you only have sleep issues and you're okay during the day, you don't have stress and all that stuff, then you would just take one sleep at night, right? So the sleep is formulated with 30 capsules, the cope is formulated with 60 for that reason. If you're using them both, the cope will actually last you 2 months because
Got
you
it.
only need one cope during the day.
Okay, I like it. Okay, guys, we have too many questions right now to be able to get to all of them, and I only have Kiran for about 2 more minutes, so I'm so sorry, but that was so interesting, Kiran. Thank you so much for that. We do have Kiran back next month. I know today's just May 1st, but we do have him back in June. Looking forward to it. Please put some love into the chat for Kiran. And please, Kiran, share the gratitude that we have with your entire team. It means so much to us that you're doing such great work and so consistently maintaining that integrity. I thank you so much on behalf of everyone here and just the people that you're impacting around the globe. This is a great message of hope. This is hope, folks. Big time hope. The power is there. The efficacy is there. Not everything's for everybody. I get that. That's not what we're saying. We're saying it's worth a try. And so many people, are coming back for more, meaning that this is what I used to say on TV when I was selling stuff. It's one thing to launch a product. It's another to have a product showing efficacy to the point of where people are reordering it month after month after month. You can do a flashy launch, but the fact that people are coming back for it and won't be without it, that is a testament to quality and an impact. So if you've ever taken a probiotic before and you never felt a difference, maybe it's because of some of the things that Kiran's been talking about today in the difference between companies. Science is leading his company. And I will never, ever bad talk another company. Like people ask me about different brands and what do we think about this and stuff. That's not the platform. This is not the platform for this. I understand your questions, but this is about what Microbiome Labs is doing to help the planet, basically. So help yourselves. Kiran, thank you so much. I'm going to let you go to your next appointment. I want to always be careful about your timing. I love you. I thank you. And I'll stay on to just do some quick chats with everybody. Mwah!
And thank you, everyone, for the wonderful comments on there. So we'll keep up the work. And, anytime Shivan invites me, I'm here. So thank you so much. Appreciate it.
Love ya. Bye. Thank you so much. That's so cool, you guys. This is what I love doing, connecting you directly to the source, whether it's Kiran Krishnan from the lab at Microbiome Labs, or we have Dr. Mark Pimentel coming up and Dr. Ali Rezai coming up after Digestive Disease Week, which is happening in just about a week, the largest gastroenterology conference in the world. That we have a tradition of having him come on and sharing his insights. So if anybody here has SIBO, please make sure you open the email where it says Dr. Pimentel on the top, because that will be his recording. It won't be until later on. I think our appointment with him is on the 18th of May, and then we'll release it shortly thereafter. I can't give you the exact date yet. Okay, you're on his site, Rob, and I don't see those last two products he covered. What is the name of them exactly? So it's ZenBiome Cope. There you go. Okay, and then Robin, it's ZenBiome Sleep. I can tell. There you go. Okay. Good. I'm glad that you're enjoying it, Marcy. That's great. Okay, good. I'm glad you see that. And that 15% off is good off of everything on the site, including the vaginal microbiome test and the BiomeFX, which is the stool test, which is also very, very high quality. So I just, that was actually not the original intention when we were able to get this 15% off from Kiran, but he just sort of grandfathered it in. So that's so cool. What's the best time to take probiotics? He talked about that. You do want to take them with food, but then you can do the Zen Cope and the Zen Sleep by themselves. Okay, he talked about that. We will be sending out the replay. So look for that later on in your email. And just to answer another question that comes up a lot is that megaspore does not overpopulate your gut. So when I was first learning about megaspore back in 2017, or spore probiotics in general, right? So that's when we was kind of launching everything. I read a blog post from a very informed, reputable, online, functional practitioner. And he really scared me about how they are known to overpopulate the microbiome, like to that it would take over. I can't remember the word right now. And so when I met Kiran, I was like, listen, is this going to take over my whole microbiome? Yada, yada. And he's like, no. And then he explained to me. Now, we have many, many other sessions with Kiran where we do talk about that, but I just wanted to mention it because a couple of people have been asking about that. Right. Don't overthink that. It's fine. I would never, ever talk to you all about something that I thought was going to take over your, microbiome in that way. Thank you so much for all your good words and for being here. I hope that you will take advantage of this if it's right for you, if it's appealing to you, if it calls to you, follow your intuition. And then what I want to say to everybody is wear it loosely, okay? Because hypervigilance and SIBO panic is something that we have a lot of experience with in this group. And I want you to get the information, and I want to have you apply your info to the learning, right, to the way that you think, because that is going to guide you and the way you intuit. And that's what's going to guide you. If you feel like this really sounds like it's different, gosh, I've tried everything else, it feels like. But my favorite question is, have you really tried everything? So that being said, breathe. Doesn't that feel better? Yes, the discount is available for Canadians, and they do ship to Canada and the Netherlands. The postage is probably going to be a bear, but they do. And right now I have Kiran scheduled. I'll just give you, it's not in stone. Okay, so I'm going to disclaim it. It's for June 5th. Literally last year, we booked him through the whole year. Do I have him in October? Yep. We booked him through the whole year. I don't think it's like November, December, because we knew that you all had more questions. And we also knew that he was super busy doing all kinds of great things. Clarissa, are you telling me that Canada international shipping is just $15? That's amazing. That's fantastic. Okay. So Joe is asking, how should megasporebiotic be timed with antimicrobials? I've heard both, but from Dr. Allison C. Becker, she does often say to take them together. And then she quotes the person that did that research, and I can't remember that person's name, but taking them together, which sounds counterintuitive, right? Often helps with the efficacy of the antimicrobial and antibiotic. Not a doctor. I'm just repeating what I think I remember she said. Okay, let's see. Oh yeah, Sharon, that sounds good. Is Megagenesis a megaspore product? I don't know, Darlene. I don't know. Clarissa, what do you think? Sandra, you're not able to register for some reason with the link. I don't know. Okay, so Microbiome Labs has no relationship with SIBO SOS per se. So if you have an email at SIBO SOS, like our internal stuff, and you bought SIBO Recovery Roadmap, that's so weird. Two people are saying that they're saying that the email is wrong. Why don't you email us? Okay, we may have just crashed the site, you guys. That'd be a personal point of pride. Larissa, can you get over there? Can you get in? That's funny. Not really. It's not really funny. Try that link that Clarissa just popped into the chat. Okay, Laura, one of them does have B vitamins in it. So that is the ZenCope. So I understand why you're asking, but this is interesting. I can only have hydroxycobalamin for B vitamin. And that is the kind that he chose to put in here, which is the one that is very, very easy on most people. So whoever's doing the genetics for your son, go to the site when it's not getting crashed by us and ask them if these two types of B vitamins are okay, because they're different forms of B6. There's different forms of B12. So that's what I would ask. The sleep might be fantastic for him though. Okay. I don't know about that. Good luck, Laura. I'll be thinking about you for sure. I know, Candace, I didn't get a chance to ask him that. I'm so sorry. I'm gonna, I got in and got registered, Luanda saying, huh, Jennifer, did you reach out to Microbiome customer service? Yale, you're on the Yale, you are on an iPhone and it wouldn't let you take it. It wouldn't do it. That's weird. Is SIBO SOS already populated in there? Do you have to be a patient to register first? So you can, so there is the magic Microbiome Labs customer service email. So it is populated in there depending on the link that you use. And when you email them, please tell them that we sent you. Okay. On the computer it worked. Let's see. Do you have to be a patient? So you have to register as a patient first and use code SIBO SOS. Yep. Hang in there with it. How long do you have to take Megaspore? Oh, Marilyn, that's a great question. Can you make yogurt with Megaspore probiotic? I'm going to ask him that. How long to take it? So Karen and I have talked about this on multiple occasions because that's a really great question. A lot of people ask it. Our ancestors populated their microbiome with the dirt from the potatoes and the tubers and the carrots every time they went to the garden to make their food. So that's the way I personally see it. You may decide to take a break and then come back to it. Follow your intuition. But I'm happy to take it long term because I'm not going out to my garden and plucking, the veggies. And frankly, I clean my vegetables and fruits within an inch of their lives because I'm not afraid, but I'm concerned about food poisoning. And especially because I mainly do organic produce, and good news is there are no pesticides. Bad news, there's sometimes bugs on them. So please apply that to the way that you think about it. Okay, we're definitely going to send out an email, and we'll definitely give you the additional information and the replay and the link to order and the coupon code and all that. Okay, I just wanted to mention that all to you. And if you can't swallow capsules, you might want to think about the gummies, which are vegan, by the way, because a lot of gummies are made with gelatin. And yes, Deborah, you can still open the capsule of Megaspore if you want to sprinkle it in your applesauce, whatever, your food, it doesn't matter. And it's totally fine. Kiran has talked about that for ages. And when you're starting Megaspore, start slowly. Start slowly. This is one of those things where we think, oh, it looks like vitamin C. It's just a capsule. How powerful could it be? I just bought it. I didn't have to have a prescription for it. It's very, very powerful. As far as those of you with SIBO, yes, as far as taking it for maintenance, I wanted to address that about the carrots and the potatoes and stuff. As far as SIBO goes, this is your personal snowflake microbiome. So what Dr. Alison Seebecker, world-renowned SIBO expert, says is if you're taking it before treatment and even during treatment, and you get a great result from your treatment, cool, keep going with it. But she doesn't like it when people haven't been doing it, do SIBO treatment, and then afterwards add the probiotic after you just worked so hard to get that, that SIBO breath test negative. So I like to tell people, start it beforehand, do it during treatment, and see how you're doing. So try to start it a while before doing treatment. But on the other hand, if you have Xifaxan and neomycin right there and you're ready to go for treatment, then don't let anything stop you. So you have to use your discernment. You have to use your common sense. What's going to work for you? Okay. And I understand, Charlotte, while you're asking that question, Megaspore is a gatekeeper. It's a keystone. So it helps to regulate and balance the players in the microbiome. So that's the best way to think about it. That's why we do this, is to teach people how to think. That is part of our goal here. And I always like to give you a coupon whenever I can, because I like to save money, and this is an expensive dealio to get well, and stuff's expensive. Barb, have you been working at all with Sinclair Canale? She's so good at liver and gallbladder stuff. I think you have, but I just wanted to check. And I know this person saying anonymous attendee. I know you're talking about that Cedar Sinai low fermentation diet. Sue, I take it with food. We already talked about that. Already, the paraprobiotics from a healthy gut are just different. And Let's see. Megaspore and Floristore, different strains. What's the difference between those two? I think Carol, we answered that question. Okay. Clara, we have asked him about the, I don't know if you were here last time and asked this question again, about histamine and mast cells. He has not seen a problem with that. He often has talked about how it actually helps them. Everybody's different. I'm just telling you what he said last time. Megagenesis, I don't think I think someone posted it's not a megaspore product. And there you go. Okay. Whew. What are you saying, Clarissa, about planning your lymphatic rescue? What's that for? I'm not really sure what that's for. Okay. Oops. Okay. That's for something else we're doing. All right. Thank you, Marian. Thank you all so much for being here. And thanks, Robin. I really appreciate it. This really is the mission that I've so gladly taken on to help all of you. But I want you to balance it. Omega Genesis is on the site. Okay. Sorry. It is L. reuteri strain. Okay. Thanks, Kay Nelson. I appreciate that. All right. Then I stand corrected. I appreciate that. I want to find out about the yogurt business and if we can make megaspore yogurt. Wouldn't that be cool? That would be so. Oh, I love that idea. I've got yogurt brewing downstairs right now. I know, right? Don't go crazy. I got a blog post coming out too about the yogurt. I'll work on it. I'll try to find out if that's the case. Okay, I'm going to wrap it up. Thank you very much for being here. This one went long. We'll see you June 5th, assuming no changes happen. Watch your email for the replay and for further invitations. And I don't usually announce the next one at this one because if it's subject to change. But on the other hand, if I do the disclaimer, it's subject to change, then you know. Okay, thanks all. Bless you all. Thank you. I'm glad you were here. Dina and Alice and Joe and Sharon and Laura and Candace and Tess and Virginia and Yael and Clarissa. Blessings all around. What I was trying to say is wear it loosely, right? No SIBO panic, no anxiety panic. If you're freaking out about stuff, try the ZenBiome Cope. Get with your practitioner. Play the summits and the podcasts in the background. You know, sometimes just having it around is super helpful. Sometimes you need to take a break too, but just casually and slowly but surely, it will integrate. I understand that sometimes you need to be super, super focused too, smooches. But what I'm trying to say is don't panic. There is hope. There are answers. And inch by inch, it's a cinch. Okay. With that, I'll see you next time. Thanks all. Bye-bye.